In last episode, 2 virions tried to infect a goblet cell and were obliterated in the attempt. The goblet cell was able to deal with the 2 virions without any problem. But the cell has an antiviral measure to prevent any more virions from infected neighboring cells, the interferons. Interferons are messenger proteins that cells release after detecting the presence of viruses. They serve as alarms that alert neighboring cells to impending danger. In the event that a cell has already been infected by a virus, interferons also serve to rescue it and cure it of the infection, being of great help to prevent viruses from colonizing a tissue. Regulators that were activated by toll-like receptors and Rig-like receptors bind to importins, and these bind to the nuclear pore complex to be imported into the nucleus. Once in the nucleus, the JNK protein phosphorylates the regulator c-Jun, and the protein p38 phosphorylates the regulator ATF2. c-Jun and ATF2 bind to form homodimers. Regulators IRF3 and IRF7 form heterodimers, but can also combine with each other to form homodimers. And the nuclear factor kappa beta is made up of p65 and p50. Later, these regulators bind weakly to DNA and begin to travel through it, sliding down their spirals in search of the gene that codes for interferon proteins. The gene is inactivated on chromosome 9 by histones that guard and protect the information. At the beginning of the gene there are 4 regions to which the regulators bind strongly to activate it, and we call them cis regulatory sequences. Regulators p65 and p50 reach the second region and bind tightly. Regulators IRF3 and IRF7 bind to regions 1 and 3, and c-Jun and ATF2 bind to region 4. When the 4 regions are occupied by regulators, they form a complex called enhantiosome. This complex binds with the protein GCN5, an acetyltransferase that adds acetyl groups to the tails of histones H3 and H4. Then a kinase protein also binds to this complex and adds phosphates to the modified H3 histone tails. Once the H3 histones are acetylated and phosphorylated, the GCN5 protein adds an additional acetyl group to them. Once the H3 histones have been modified, the BAF complex binds to them. This complex displaces histones to uncover a sequence called TATA box and activate the gene for interferons. Transcription factor 2D binds to histone H3, and this in turn is bound to TBP, which binds to the TATA sequence in DNA. At the same time, the P300 protein also binds the enhantiosome complex. This protein helps attract RNA polymerase II and transcriptions factors, so they bind faster to genes and accelerate the reaction. RNA polymerase II begins to polymerize an RNA strand that complements the DNA information. Some proteins add a cap to the RNA on its prime 5 side. Then, different proteins bind to the tail of RNA polymerase II, and these bind to RNA sequences that divide introns from exons. Spliceosome proteins bind to these proteins and separates introns from exons. Finally, other proteins add a repetitive series of adenines, called poly-A tail. At the end, a mature messenger RNA is synthesized and is subsequently transported out of the nucleus to the cell cytoplasm. Here the messenger RNA meets the ribosomes. First, the 40S subunit binds to the mRNA and moves forward until it finds an AUG sequence and binds tightly. Then the 60S subunit binds to it and begins to translate the information from RNA to protein. The ribosome binds to proteins in the membrane of the endoplasmic reticulum and introduces the interferon proteins inside it. The new protein begins to fold as it enters the endoplasmic reticulum. Here oligosaccharides are added and chaperone proteins help the protein fold correctly. Once the protein folds completely, it takes its 3-dimensional form and binds to proteins in the membrane of the endoplasmic reticulum that form vesicles and transport interferon proteins to the Golgi apparatus. Here, the interferon oligosaccharides are modified and the protein maturation ends. Once mature, it is transported in vesicles by motor proteins to the cell membrane, where it fuses with it to be expelled to the outside. Interferons disperse in the extracellular fluid and reach neighboring cells, where they bind to interferon receptors. After binding, the receptors phosphorylate each other by Janus kinase proteins. Once phosphorylized, they bind to STATS1 and STATS2 proteins, which are also activated by phosphorylation. Once activated, the IRF9 protein binds to STATS2 and the protein complex is imported into the nucleus. In the nucleus, STATS regulators activate a set of up to 300 genes, which drastically change the metabolism of the cell, making it an hostile hell environment for viruses. We call this metabolic state the antiviral cellular mode. In this mode, the defenses in the cell membrane are reinforced through the expression of transmembrane proteins called aphidim. These prevent the membrane fusion of the virus with the cell, increasing the viscosity of the lipid bilayer. It also increases the expression of tetherine proteins, which anchor to the membranes of viruses, disabling their escape. In the antiviral mode, protein production is also stopped and cytoplasmic RNA is degraded, interrupting the viral cycle of any virus. All cytoplasmic messenger RNA is degraded by the expression of endonucleases that cut RNAs in two. The PKR protein phosphorylizes the ELF2 initiation factor, inhibiting its function, essential for the ribosome to find the AUG initiation sequence in messenger RNA. Proteins that attract cells of the immune system, such as dendritic cells, macrophages, and neutrophils, are also expressed. They also attract natural killer cells, which induce cell death on infected cells. These and many other metabolic modifications prevent the virus from infecting and replicating within the cell. But at the same time, it affects the normal function of the cell, affecting the normal physiological functions in that tissue. The other neighboring cells go into antiviral mode, increasing their defenses. This prevents other virions from infecting the tissue of the nose, and the organism ends up eliminating the remaining virions. This case represents a person who takes precautions by keeping his distance to a minimum of 2 meters. 5 minutes later, the bus waiting for the lady arrives at the stop. She gets on and sits in the middle. The bus has air conditioning and closed windows since it is a very hot day. This creates a current of air that circulates the bus continuously, and with each breath the microdroplets in the air increase and increase. The most affected people are those who are directly in front of the lady. But the bus windows are closed and the microdroplets slowly disperse equally throughout the bus. In this scenario, the amount of microdroplets is much greater. Each breath is like a microscopic rain inside the nose and throat. Thousands and thousands of viruses pass the mucus barrier and go towards the cells. In the next episode, we will begin to see the mechanisms used by the virus to overcome antiviral defenses and how it manages to infect and take over cellular metabolism.